is Dr. Lisa Rothman, who will be speaking about soybean disease challenges.
Right, thank you very much for the opportunity to be here today. Mostly, I have to apologize, my profile photo I see was in the sunflower fields. I'll have to prepare next time to make sure that they're in the soybean field. So thank you so much for the opportunity to be able to share my little bit of a story on soybean diseases. So if we look at the situation with soybean in South Africa, we see that there's a trend that's increasing over the last while, like Joseph said earlier. And what's important to remember here is that the FAO have stated that 3.3% on average soybean make up our caloric intake. And that is actually quite significant if you think about the contribution that this crop makes and is continuing to make over time. Now, I put in the provincial distributions because this becomes relevant when we look at where soybeans are being produced and where they will be produced in the future. And why is this relevant to plant pathology, you may be asking? Well, our plant health is so important to understanding the disease triangle and how this fits in. And there are predictions that soybean production will be expanding into marginal areas. That means that we need to have an awareness of this giant, the disease giants. And so let's think about soybean as the host crop for a minute. Soybean were introduced into South Africa in 1903, and we've really seen an expansion over the last 20 years. But along with the soybean, we have this very dynamic environment that is occurring. It's not only the agricultural environment, but also the climate environment that's associated with the expansion of this crop. And not only in South Africa, but globally. There's also the socio-economic dynamic that's happening in this environment. And that's critical because when we look at the biotic disease-causing organisms, so these are living infectious organisms, we need to be aware of where are they found? Where are they more prevalent? What can we do to suppress them? And when I was being trained as a plant pathologist, my professor, Professor Neil McLaren, would say, think of the disease triangle like a potjie. And South Africans love a good potjie. If you have the 3 feet at the bottom and you pull one out, that's when the potjie can topple over. And understanding the disease triangle will allow us then to topple certain diseases that we need to be able to manage. So according to Savary et al. 2019, there are approximately 25 significant biotic disease-causing organisms and pests associated with soybean. So what I want to do today is just go through some of the research that's been conducted in South Africa and what should we keep an eye out for going forward, seeing that soybean production is going to continue to increase. So I have to say thank you very much to the Protein Research Foundation and the Oilseeds Advisory Committee for having the archived research available on their website. And what I've done is I've taken that information and I've created a timeline. So first of all, I have to say a little caveat here. If you've worked with industry or private or with the NRF, you may not be present on this slideshow, and I'd love for you to contact me so that we can add that onto this. But what I've done is I've summarized the principal investigators from projects on the PRF research archive. And these archives stretch all the way back to 1986. So when I first went to look, I thought, wow, this is incredible. Since before my time, there's been really great research that really inspired me when I started working on this presentation. So by no means am I an expert on all the soybean diseases, but I'd like to thank everyone else that's contributed for me to be able to stand here today on the shoulders of giants. So if we go through the research that's been conducted, there's been a focus on soybean mosaic virus, nematode resistance and chemical control. Then we look again at the importance of identifying viruses, and then we start to move into the world of soybean rust and Sclerotinia stem rot. And as we get into the the 2010s, we see that the nematode research continues as a focus, as well as soybean soil-borne diseases, charcoal rot, and the root knot nematode. So I do apologize for some of the placements being out of place, but my presentation is available online, and I will share that with you all later. So if we look at viruses, we can start to see that there are two critical viruses that have been associated with soybeans. The first was found in 1961 in 3 provinces in South Africa. And this doesn't necessarily affect the yield, but it affects the quality of the seed. And this is very important for our producers because it comes down to how are the seed being graded, and also this transmission of this virus is with seed, but also it has over 30 different aphid species that are known to have a non-persistent transmission. So if you see the little triangle with the time next to it, that is an indicator of emerging diseases that we need to keep an eye on going through this presentation and obviously in the field. So there's a really lovely paper that has explained viruses in South Africa from 1985 to 2012. It would be great if we could have an updated one for 2021, 2022, a 10-year later kind of perspective. And of these that were detected, we see the African cassava mosaic virus has actually been reported to be a virus of soybean. The others are not on soybean, I dare say yet, but haven't been reported yet. So soybean rust is soybean rust is caused by an obligate parasite, Pacospora pacaraisi, and it was first reported in 2000 in KwaZulu-Natal on soybean, and in 2004 on dry bean. And it's a very interesting timeline, from when it was first reported in 1903 all the way up until it made landfall in the USA in 2004. And what we as South Africans can be so proud of in our agricultural industry is that in 2001, when soybean rust made landfall in South Africa, from being present in Africa from the mid-1990s, immediately there was a soybean rust workshop that was initiated to look at how we can mitigate and manage this. Now, if we look particularly at soybean rust in America, it made landfall in Alabama in 2004. And in 2005, they initiated a program to assist producers with an integrated pest management for controlling soybean rust. Initially, in 2002, there were only 5 registered fungicides available in the USA. And in 2010, there were over 70. Now, this becomes important, as you will see just now. So we have the PIPE 2021 image in front of you. And this is to show the capacity that America has for daily scouting of sentinel plots. Then we have Brazil that has a different management strategy, where they have soybean-free periods. And this becomes important because the pathogen survives on hosts throughout the year. So being able to delay that epidemic is really critical for reducing the impact of soybean rust. So in South Africa, we've had many sentinel plots and reports of soybean rust. And then we will see that in 2014, there were 16 registered fungicides, and since then, there have been 4 additional actives or fungicides added to that. In Brazil, there are over 120 registered fungicides for soybean rust. Only 3 of those have over 70% efficiency. So it goes to show you can have as many fungicides as you want registered, but what is the efficiency of those fungicides to protect or mitigate the losses? And what is the cost effectiveness of this? So we have a airborne disease like rust. Let's look a little bit below the ground. Something that is caused by Fusarium species. And the soybean sudden death syndrome, or SDS, is something that was first reported in 2013 in South Africa, as well as the Fusarium brasiliense. But there is an unknown Fusarium species that is associated with this disease complex. And we see the symptoms on the leaves, but it is associated with the roots. And if we go and sample the leaves, the pathogen has not been detected in the leaf samples yet, but only in the roots. So this is a very fascinating disease or pathosystem to work with. Now, researchers in the United States have actually got this really cool early detection system that they've been working on. And this is from a colleague that I've met, via the internet. It's just such a wonderful thing that we actually have this ability to, meet people without having to physically meet them. I hope that I get to meet him in person one day. And through remote sensing and machine learning, they've actually been able to use drones to go and look at how well can the drones predict the disease prevalence or the severity? And they have people scouting in the field to then go and look at how reliable is this data. And they've worked out that it's reliable enough to allow for this early scouting. And why this is critical is because the symptoms are on the leaves, but the pathogen is living under the soil on the roots and can affect soybean anytime from planting to harvest. This gives leverage to the producer to ensure that it can be detected earlier than what it is visibly seen. So if we look at charcoal rot and southern blight, in 2014, Dr. Lisa Rothman said that he believes that Macrophamina fasciolina, the causal agent of charcoal rot, is one of the most important pathogens associated with the drying down of soybean. And I must say, I think with climate change happening, and when we look at how the climate or rainfall patterns are changing, and perhaps also, the frequency is becoming greater or less, and the amount of rainfall, this is something that we should definitely be keeping an eye on. Southern blight is caused by Sclerotium rolfsii, and it was first reported in South Africa in 1926, but on tobacco. Now, we know that it occurs on soybean. I took the picture that doesn't have the circle around it, and often this pathogen gets confused with Sclerotinia sclerotiorum. But it's easy enough to distinguish it when you're aware of the signs of this disease. Now, if you see in the circle, there are small spherical sclerotia. Now, the sclerotia are the survival structures, and these survival structures allow us to distinguish between Sclerotium rolfsii and Sclerotinia sclerotiorum. So Sclerotinia sclerotiorum is a passion of mine, and I may not be an expert on all the diseases, but this is something that I'm hoping to grow as an expert in going forward. So Sclerotinia sclerotiorum is the causal agent of Sclerotinia stem rot of soybean, and it has over 500 host crops or host plants, not just crops, and in 1979 it was first reported on soybean in Mpumalanga. Now, let's look at the signs of this pathogen. There are the white, fluffy mass of mycelium, and as the season continues, this mycelium can dry, melanize, and harden, and it actually shapes and forms sclerotia. These are survival structures that can survive very long in the soil. Now, as you can see in the picture, the sclerotia have actually formed inside the soybean pod. And it hasn't replaced the pod, it has just formed alongside with it. So we need to keep in mind that the persistence of this pathogen and the pathogens that I've spoken about lies in their ability to survive. Soybean rust has the ability to survive on their alternate hosts and alternative hosts. We have the sudden death syndrome pathogen complex that's known to survive in soil and on stubble. And then we have Macrophamina and we have Sclerotium, which both produce sclerotia or microsclerotia. And the survival of these pathogens is where we need to be able to interrupt the life cycle and mitigate any losses that can be associated with them. During my PhD study, I had the privilege to work with the Crop Estimates Committee, who collected 10 years of data on Sclerotinia sclerotiorum surveys collected in South Africa. And what we did was we wanted to have a look at what was the severity that producers were observing in their fields, and how often were they observing these severities. So if we actually were privileged enough to have both the soybean and sunflower results, but today I'm just going to be presenting on the soybean. So if we have a look at the percentages, so the percentages above the bar are representing the severity or intensity of Sclerotinia reported in the soybean fields. And this percentage is reference to the hectares that are diseased in context with the area that was planted. And if we look at the frequency, that is the amount or the volume of producers that were responding to the type of severity class that was seen. So it is of a great relief for us to see, over 60% of producers were observing less than 1% severity. However, even though there's only 1%, that 1% carries over into a next season because of the sclerotia that can form and persist in the soil for up to 12 years. So what about light, moderate? Yes, these are nearing 10% frequencies, but have below 40% disease severity. My concern is for the producers that are seeing severe and very severe stem rot in their fields, because the available chemistries is very limited, and it is important for us to look forward to how can we get registered active ingredients, yes, but how efficient are they? So if we have to talk about Sclerotinia a little bit in detail, let's talk about the population. So some research that has started with my predecessor, Professor Neil McLaren, and Dr. Krishna Stein, started with looking at how different are the different isolates collected of Sclerotinia sclerotiorum across South Africa and from different host crops. This research is now continued with Carmen Meyer and Dr. Adrian Menard-Ontong, as well as Dr. Stein. And there are currently 650 isolates that have been molecularly analyzed, and to look at how similar or how different are butternut isolates from the sunflower isolates or soybean isolates. And they've been collected across South Africa. There are an additional 650 isolates that are going to be included. And the value of this research is that we can say that a Sclerotinia sclerotiorum isolate that infects butternut has the same capacity to infect soybean, or cabbage, or carrots, or sunflower, or squash. And the wide host range is what makes Sclerotinia so successful. And that's what's really important to remember about Sclerotinia sclerotiorum. There is no forma specialis like they have for the rust pathogens. One Sclerotinia can cause a disease on over 500 host plants. There are even some weeds that are associated with Sclerotinia sclerotiorum. So in South Africa, we have legislation, and we are one of the very few countries in the world to have legislation against the presence of sclerotia in seed. And this is great. And Sansoil have actually improved this to make sure that certified seed are not allowed, that the maximum level is 0.2%. So I think that we can commend and really look up to our industry for taking this seriously, because there are only 3 other countries, Brazil, Australia and Canada, that have restrictions, and Brazil's restrictions they do not tolerate any pathogen or Sclerotinia sclerotiorum in their seed. Now the question we receive often is, can Sclerotinia sclerotiorum mycelium survive inside the seed? And unfortunately the reality is, yes, it can. So if we look at the images on the top, we have a semi-selective media that is preferential to the growth of this pathogen. And why this is really cool for me is that when you plate seed out, it makes a yellow halo. And this yellow halo is an early indicator of the presence of the pathogen. This being said, it is usually only diseased seed, seed that you can visually see is diseased, that will have Sclerotinia sclerotiorum. Healthy seed or non-symptomatic seed does not contain Sclerotinia sclerotiorum. And we have an honors project currently running, looking at the volume of seed and the prevalence of Sclerotinia sclerotiorem in the seed. And I think that this is going to be vital information for producers and for us as plant pathologists to keep in mind when we're looking at potential seed treatments. The graph that you can see in front of you is the sclerotia per sample that was collected from different silos across South Africa. And this information is also, all the data are collected by Southern African grain laboratories, and the funding is provided by OPORT and the OAC. So I'm really grateful that this is publicly available. And what we can see is that most of the samples collected, ranging from 100 to 150 samples, are below all of them are below the legislative limit, and only 2 of them are above the 0.2%. And this is an indication, potentially, of what is happening in the soils around the silos in South Africa. So this gives me hope also, because if we have producers using farm-saved seed, that if they can clean their seed and sieve out sclerotia, that we know that they can reduce that prevalence of sclerotia in their seed. I took to Twitter and I asked some of my plant pathology colleagues if they had to consider, what important global emerging pathogenic threats are there to soybean production. And I actually got quite a few votes in a 36-hour space of time. 72 votes. I do not have many followers, so this was really great response. And even better than the votes, I got really interesting communications and discussions. So if we look at anthracnose, it was the first and most voted for emerging soybean disease. Now, the focus of research has been on Calototricum truncatum, but at least 9 other species and other complexes are associated with this disease. As you can see, it affects all parts of the plant. All right, what about other? Now, these were some of the comments and discussions that came up. Taproot decline, target spot, frog eye leaf spot. If we look at taproot decline, only in 2017 did they first identify the pathogen Xylaria necrophora as the pathogen. So there are so many diseases that we're still unaware of. Then we have target spot and frog eye leaf spot. These are foliar pathogens that are reducing yield through reducing the photosynthetic capacity, and they can also cause early defoliation. Then there was talk of Pythium, which are not real fungi, not a fungus. They're part of the oomycetes, and Phytophthora, as well as bacterial diseases. One of the issues that came up was monoculture. And sure, Ed Sikora says this is a threat as a practice leading to disease problems in soybeans. Nematodes and many other diseases would like that. Now, I'm pretty sure many of you thought I'd omitted the most important biotic pathogen, and I have not. Nematodes are one of our most prevalent and noteworthy foes or enemies of soybean. And their persistence in the soil is something that really needs to be monitored and taken account of on a regular basis. Now, root knot nematode was first detected in 1959. Now, I've said and company. There are many of these species available. Then we have the lesion nematode. In South Africa, we do not have the heterodieroglycines or the cis nematode present yet. They also have a wide host range, as well as a weed host list that they can survive on. If we look, agriculture is not static and in isolation, and many of these fungal pathogens are actually potentially working in unison with the root knot nematode and lesion nematode, either making them more susceptible or increasing the severity. But we need to recognize and acknowledge the hard work that the nematode researchers in South Africa have done. Yes, they are champions, but with this championship comes a burden. And that burden may also be the fact that South African nematology is not just for South Africa, but the whole of Africa. And we need to assist them and carry that burden alongside with them. So this is just a small token to say, I think that South African researchers need to put a focus on nematology in the plant pathology world. And I hope that this is something that we can encourage in our different plant pathology departments and divisions at our academic and public institutions. So I've done a little survey of some literature of the top soybean producing countries, reported diseases globally, which are not ranked, and then in the southern United States. And on top of those lists, from 1994, remains the soybean cyst nematode, which we do not have in South Africa, I say yet. And this is something that we need to make sure that this remains outside of South African borders. But if we go and look further down, you can see how Sclerotinia stem rot has climbed the ranks of soybean diseases. And globally, this is considered the second most important disease on soybean, followed by soybean rust, Cercospora leaf blight, and Septoria brown spot. So we also need to keep in mind that we need to move back and beyond the pathogen. And this is the McLaren philosophy that I've been taught. It's not just about the cultivation, but what happens afterwards. As we have an increasing middle class, more people are eating meat. That means more soy is being used for livestock feed. And some of these pathogens have the capacity to produce mycotoxins. These are toxic secondary metabolites that can be caused by the Fusarium species and the Alternaria species. Now, the Fusarium species are regulated by government, and there are thresholds or limits on this. However, the Alternaria species are not monitored yet, and this is something, or legislated, they may be monitored, but there is no legislation against them yet. And this is something that should be regulated. And I know that the Southern African grain labs are responsible, or not responsible, but they are looking at mycotoxins in the different grain products. So overall, we need to be able to see eye to eye, and that is actually what's come up in literature. We need to have continuous surveying and monitoring. We actually need to increase our capacity in plant health. We need to ensure that there's a focus on disease-free planting material, looking at alternative sources of resistance for our planting material. Look at the available pesticides and their sensitivity and efficiency. Ultimately, this needs to be integrated for the producer. But in South Africa, we need to have a little closer look at the yield loss assessments and the economics and the of each of these, and how valuable or how worth it for the producer is the use of pesticides. We have to have a long-term focus and have an awareness and education of plant health and our intimate relationship with plant health, because plant health is our health. So this is just a big picture of the tonnage and the years. We can see that there's a flow. Right? So some years the yield or the tonnage is lower than average, and some years it's higher. But we need to be able to know which pathogens are causing the most devastation so we can control those. And the things that we cannot control, we can then leverage, because we've controlled what we know. So this is just an acknowledgement of all the hard work that the people that have worked in this presentation or the references that I've used, and I just want to say, arise, sirs and dames, for the service to plant health in South Africa. Thank you for contributing and for giving me the passion to be able to be here today. So if you would like to reach out to me, these are my contact details. My full presentation is available if you scan that QR code. It's also citable, and if you would like to contribute to the written work of this, I have Prof. Driekie Fourie that's going to be joining me. Please feel free to contact me. I just want to say thank you again for the wonderful opportunity, and I hope that you have a few questions, because I think that's where we learn so much from one another. So thank you.